Hi, the name's Gold, and I'm just here to talk about like some stuff I learned about I'm just starting to go through my biology A-level textbook over my holidays. I know, enjoy your holidays, girl. But yeah, basically, there's some topic that I've gone through that is quite interesting, and I just want to try to explain it the best I can to whoever's watching. Anyway, so basically we're gonna talk about how microscope works and preparation, how you prepare substances or substances for them to be viewed through the microscope. So we know that microscopes were created by Robert Hooke, who basically saw a cork and sliced it into thin slices and saw that it looked like a monk's chamber back in those days. I don't know what those days are. And basically he kind of was the first to view cells and name them cells because he reminded them of the monks thingy. Anyways. So in a microscope, there's two lenses, and them two lenses are the objective and the eyepiece lens. The objective lens is basically used to magnify what's already in the slide. So there's different, they can be different magnification. So if you want it bigger, you can turn it to a magnification in the eyepiece that you want. If you want it smaller a little bit, you can turn it to whatever magnification you want. We also have the eyepiece lens, which is basically where you will be able to view the cells as big as you want it, because you already put an objective lens in the bottom, so you can see exactly what you want. Okay, another thing is that for a light microscope to work, you need to have light coming from either beneath or above. Some people like to go fancy and go above, but we're gonna talk about how it goes from beneath, because when light goes from beneath, it kind of through different refraction, because goes from beneath, goes through glass, and then it goes through air, and it's just different density, so it makes it much more, it makes it much more easier to view the cells, and it just makes it easier to view the cells. So when it comes to sample preparation, we have 4 different sample preparation modes. We have the dry mode, which is basically where you take dry substances and you make sure it's really tiny pieces of that substances, because let's say for example hair strand. You take a hair strand, you put it on top of the slide, and you put a cover slip on top of it, and then you view it through the eyepiece, and that's pretty much it. If you want it bigger, you turn the objective lens to whatever magnification you want. Another thingy is basically the wet mount. The wet mount is basically about wet stuff, technically. For example, blood. You take a droplet of blood and you put it on top of a slide, and you don't just put the cover slip on top of it like that, because there could be bubbles and that's not good for the experiment. So you put the cover slip on top of the droplet that's on top of the slide through an angle, so to avoid any bubbles, and you view it to the eyepiece. And if you want to change the magnification, you got objective lens. Another is a squash slide. Basically, it's just to view tissues. Okay, let me give you an example, because I don't really know much about this, but it's basically squashing, like it says. So you take let's say for example a root tip, you put it on top of a slide, and you put the cover slip on top of it, and you gently squash it. If you use too much force, the cover slip could break, and that's not good for investigation, is it now? Because it's just a whole thing I can't. Okay, squash slides, you squash it and you view it through the eyepiece, and you see what you want to see. Another thing is a smear slide. This one, you can I think you use it mostly for wet stuff. I don't really know that. I have not, because I've not gone that deep into the book, but it's just where I am, like right now. So anyways, the smear slide is basically, you take, like, let's say for example, a droplet of water that's like contaminated or something, you put it, and then you use the cover slip to gently smear like the water across the slide. And then you put a cover slip on top of it, then you view it through the eyepiece, and you can change the navigation if you want. Yeah. Another thing that we can talk about is using staining. Basically, it's putting colors in cells, because cells don't have colors. They're literally transparent due to stuff like cortisol. I don't think I can pronounce or spell that correctly. Cortisol. CHOROSOL. Yeah, I think that's it. You put that, and that's pretty much it. Cortisol makes it transparent, and other substances in the cell makes it more transparent. So you put staining, like maybe iodine or any other staining